we'll have a chance again when we end this session for Q&A about clinical trials and things. So if you have questions, we'll be able to address those as well. So the next discussion is going to be led by Dr. Colin Pritchard, who's an associate professor at the University of Washington in laboratory medicine. He is truly an international leader in developing molecular diagnostics. So you heard a bit about the genomic analysis, the circulating tumor cells, the ctDNA. So Colin is a person who actually puts that into practice for how you would use it on a day-to-day basis. So Colin, welcome.
It's really an honor to be here. I'm probably maybe the only pathologist or laboratory medicine doctor talking to you today. So we have the privilege of seeing the specimens, but not the patients as often. Although I have gone through medical school, I have actually talked to patients in my life. So with that caveat, all right. So what I do is more around the sort of genomics and the molecular dissection of these tumors. So we're looking at the tissues and we're trying to figure out what went wrong in the cancer. And we are lucky now that we have an array of drugs that are targeted to certain problems in the cancer. So I like the analogy I like to give for the targeted therapies is it's the Achilles heel. So we're trying to identify that Achilles heel of the cancer. These are sometimes called driver mutations, or you'll hear other terms for them, but they're the things that are making the cancer go. And now we have an array of drugs that if we can identify that thing that's making the cancer go, we can line it up with a drug that will make it stop. So that's the basic idea. We're still relatively early on in prostate cancer, so this research is critical, and particularly the IPCR has been really critically important for driving this field. So we're really thankful for that support. So you may have heard the term precision medicine or personalized medicine. The idea here is that, traditionally when we've done research to look at new therapies, we've taken whole groups of people and tried a drug, and a whole other group of people not tried a drug. And we haven't really looked at their cancer itself other than maybe the sort of basic histology, that's what it looks like under the microscope. So we might look at like the Gleason score or things like that, but we haven't looked more deeply in the past of what really makes that cancer unique in terms of how we do these treatments. So that's the difference between population-based versus precision, is that in population-based, which is the sort of traditional approach, we'll make treatments based on what the pathology says, maybe the stage, that is it spread throughout the body or is it localized? But that's pretty much it. And this sort of newer way of doing trials and thinking about how to sort of tailor the right drug to the right patient, the precision approach, would be to look at the tumor tissue, do some kind of molecular test, the kinds of tests that I do and develop, and then detect those drivers, those things that are making the cancer go, and then really try to sort of dissect that group into many subgroups and identify the drugs that might be best for that specific patient. And those groups might be small. I mean, it may be that only 1% of people with this kind of prostate cancer have this particular alteration, and yet those 1% may be very highly likely to respond to a drug. And so if you think about that 1%, and let's say we did it the traditional way where you looked at the whole group, well, you might not see any effect of the drug, right? Because it might be just that 1%. So you can imagine if you just treated everyone with it, it might be you didn't see any benefit. But now that we can see, oh, well, actually there is benefit, it may be in a small percentage, but if we can identify that percentage, we can really benefit them. And the more of these targets we identify, the more those 1% start adding up to 100% hopefully eventually. So this is the idea. So a lot of this is driven by technology. So have people in the audience heard of next generation sequencing? Is that okay? So I've seen 2 or 3 hands. Yeah, it's that's good. That's actually when I gave this presentation a few years ago. I think there was 0. So this is a technology that's really driving the ability to do this. So traditionally we've had the ability to sequence DNA at 1 gene at a time, but going beyond that became kind of expensive and took a long time. So it took us many years to sequence the first human genome. and now we can sequence a human genome in a couple days. So we've had this sort of quantum leap in technology, and the technology is so-called next-generation sequencing. It's actually a whole host of different technologies, but the thing that unifies them is that they are very much more powerful. We can sequence all the genes in the genome now for basically the same cost that it used to cost to sequence 1 or 2 genes in the genome. So what this technology allows us to do is, for the first time, really do this molecular profiling in the clinic, and it's already being used for prostate cancer care. So again, just to emphasize the importance of this technology, this graph shows the two different sequencing methods. So the first generation sequencing, we could sequence maybe 1,000 letters of the genome. So the genome is about 3 billion letters long, and we used to be able to sequence about 1,000 or so. And now we can sequence 1 billion. So it's not an incremental increase, and this is the timeline of this is in the past few years. So these technologies were introduced in the research setting in the mid-2000s, and they've been in the clinical setting for about 3 or 4 years now. So they're relatively new, but they're really a quantum leap in technology. I mean, it's from 1,000 to 1 billion, not from 1,000 to 10,000. So that's really what's driving our ability to do this. And in some ways, the technology is sort of advanced more than what we know. So we have to keep that in mind. You know, we don't actually know enough about the genome and how to do treatments. It's not matching with our technology, but we're getting there. We're getting there very quickly. So Dr. Montgomery mentioned this study, this international study that was funded by Stand Up to Cancer and the Prostate Cancer Foundation that I think many in this room have very generously participated in. This has been a really critical effort in understanding what these drivers, what these Achilles heels are in advanced prostate cancer. So this is a complicated slide, but the point of it is just to show that there are these different Achilles heels. There's these different pathways that we've identified just recently, of which there are targeted therapies. So there's a pathway called the Wnt pathway, and a pathway called the PI3 kinase pathway, and then of course there's testosterone pathway, also known as androgen pathway, that we've heard a lot about, and many of you are familiar with those drugs, but we're identifying new ways to target that after looking at the sequencing at the DNA level. There's a pathway called cell cycle that we can target, but I think perhaps most excitingly are the DNA repair pathway alterations that we found. So again, just give context, there's many pathways, there's all kinds of these Achilles heels out there that we're now just starting to do trials and learn about. But the exciting thing about DNA repair pathway is that we actually might already have some drugs available, so particularly platinum chemotherapies. Now we need research to prove that those work, but they are already available, so they can be used without a lot more work. And there's also a group of drugs called PARP inhibitors, which Dr. Montgomery mentioned, that can also potentially target defects in this pathway. And these drugs are not yet approved for prostate cancer, so they're available on trials, but they are approved for other kinds of cancers. So it's possible that they, if we see a benefit in prostate cancer, that these could be useful too. So I think in terms of what's coming along most quickly, in terms of what we've identified in the genome and these drugs, it might be the DNA repair pathway is the most exciting. So here's some data from a phase 2 trial. So we just heard about phase 1, phase 2, phase 3, phase 4. So this is an early phase trial, but a trial looking for benefit of the drug. And this is a trial of a PARP inhibitor that was done, and it was a very small number of patients, only about 60. But it was a remarkable finding in that of the 60 patients, 16 of them benefited from the drug. And 14 of those 16 patients had these DNA repair defects when we looked at their tumor tissue and did sequencing. And of the patients that didn't benefit from the drug in this trial, only 2 of them, 2 of those more than 30. So it was a very profound effect where it really, even though it's a small number of patients and we can't use this small data set in order to, sort of think about treating patients yet, we need to do larger trials and see if this really confirms. It's still very promising early data, and it actually prompted the FDA to sort of fast-track the approval process for the drugs in prostate cancer because the preliminary data is promising. So we're excited about it. but we still need much more data in order to know if we can treat more broadly with these. So there's a lot. This is a very busy slide. Again, just highlighting all of these different targets. The DNA repair pathways can be broken down into two flavors. And this is where it gets a little bit complicated even for me to figure out. But the two flavors are DNA repair pathway defects that predict to things like platinum drugs and PARP inhibitors, which are sometimes called HR DNA repair for homologous recombination. There's another DNA repair pathway called mismatch repair, which is sometimes abbreviated MMR. That is in about 5% of prostate cancers. We don't know the exact prevalence yet. But that's also very interesting and promising for potential prediction of a different kind of therapy, immunotherapies. So we have even less data about the relationship between mismatched DNA repair defects in prostate cancer in response to immunotherapy. But again, the data we do have is very encouraging, and I think enough to warrant clinical trials very rapidly to sort of see if this really predicts. So again, these homologous recombination DNA repair pathway defects, mismatched DNA repair pathway defects, overall DNA repair, I think is very exciting in terms of early therapy in the next few years. But we also have all these other pathways that I think are a little further out in terms of years before there'll be approval, but are promising targets, and they're listed here. So what about DNA sequencing in clinical care? So I'm a clinical lab director, and we are already doing clinical testing of using genomic sequencing. What we do, though, in the clinical setting is a little bit different than these research studies that many of you are involved with, in that we usually look at only a few genes. Now, we're looking at more than one or two, and we're using the same technology, this so-called next-generation sequencing technology, but we're harnessing it just to look at, in our case, we look at a couple hundred genes. So it's still a couple hundred, but it's not the whole genome. The whole genome is about 20,000 genes. And so what we can do there is we can say, okay, these are the genes we know something about already. This is something, if we find a defect there, we can actually say something meaningful, at least a decent percentage of the time. And we can harness this technology just on those genes, and then we can sequence them again and again and again and again and get very high quality data and have very, very high accuracy of the results. So these are called gene panels. And so we offer a panel UW called UW-Oncoplex. It's that. So it's a clinical test. It's designed to identify so-called actionable alterations. These are these Achilles heels. And because of IPCR, a large part, we've optimized this for prostate cancer. So we've added a lot of targets on there that are very specific to prostate cancer, which makes us somewhat unique. This is 265 genes, and it's these are just the gene list. So through IPCR, we've been for a few years now doing a pilot study applying this clinical test, and I'm sure some of you participated in this room, and we're very thankful for that. So we're applying a clinical test, again a gene panel, but to prostate cancer to learn how this clinical testing can potentially benefit men with prostate cancer. So these are done at no charge on study, of course, and then we have a prostate cancer tumor board that's focused just on the genetics. And so most of the speakers you've heard about come to this tumor board. It's once a month. And then we talk about these findings and what went wrong in the tumor and what are the drugs that are potentially available and how could we potentially do these precision therapies. And it's really, really useful. It's especially useful for me as a pathologist who, again, I don't usually get to see the patients in the clinic, so to get to hear about, how this is really impacting and the practical considerations. Well, no, he's over here. He can't get to this trial, or just to understand all the sort of complexities. It's not just about the data; it's about what's happening to the patient. It's very useful, and so we can come to rational decisions about what might be the best course of action. So, as part of this clinical testing through this gene panel, we have made some, I think, pretty important discoveries. So Dr. Chang and Dr. Montgomery made an important discovery and have published the first I think was going to be a landmark paper in terms of responses of platinum in prostate cancer patients that specifically have defects in a gene called BRCA2 in their tumor. So it was a small study. It was only 3 patients, and I'll show you that. But what Dr. Montgomery and Dr. Chang did is they looked for the patients that had the best responses to this drug of all the patients they'd ever seen. And then they looked at the tumors of those 3 patients that had had the best response, the longest response to this drug they'd ever seen, and all 3 of them had this relatively rare defect. So again, it's not enough data for us to know we need more studies, but it was quite remarkable to see that. So that's fairly exciting that this particular defect I think is likely to be a strong predictor of response to platinum-based chemotherapy, which of course is already available. The other thing that's come out of this IPCR funded study that I think is very exciting is the mismatch repair DNA repair defects, and that is associated with something called hypermutation. So if you have defects in that pathway, there's many mutations in the tumor. The tumor's got many more defects and mutations than an average prostate cancer. And that might seem like a bad thing, but it actually turns out that might be a good thing in terms of predicting if you could respond to immunotherapy. Because if you think about the more the tumor is different than the self, the more the immune system may be able to recognize it as different from the self, and the more that an immune-based therapy might work. So that's the sort of idea, and there is emerging data that this is actually potentially going to be true. So we'll see. This is just that paper led by Dr. Chang and Dr. Montgomery on what we call exceptional responders to platinum-based therapy, all having these BRCA2 mutations in their tumors. And this is just one patient who had a partial response to an immune-based therapy that had this hypermutation due to this mismatch repair DNA repair defect. So, we've talked about this large sequencing study, the Stand Up to Cancer Prostate Cancer Foundation study that many of you have been a part of. That's been critical in identifying these Achilles heels. Talked a little bit about clinical testing that we and others do that's now available, that's also driving the research through studies that apply clinical tests. And again, these DNA repair defects, I think, are really the most exciting in the very near future in terms of allowing new therapies. So it's really an exciting time for prostate cancer. And anyway, thanks most of all to the patients and their families for your participation.